Hello. Today I'm going to talk about CRISPR and how it works. First off, we need to know what this acronym stands for and its meaning. CRISPR is short for clustered, clustered, regularly, interspaced, short, palindromic, repeats. And they are short segments of prokaryotic DNA containing short repetitions of base sequences. Every repetition is followed up by short segments of spatial DNA from past exposures to foreign DNA. So I'm going to draw a double-stranded DNA and label the spatial DNA. So here. Oops. Gotcha. And the spatial DNA is located right here in between. Some applications that involve CRISPR are genome engineering, editing, knockdown or activation, RNA editing, disease models, gene drive, biomedicine, gene function, and many more. I'm only going to talk about one of them, so let's go with editing. The type of editing that I want to talk about is called CRISPR-Cas9. CRISPR-Cas9 is a genome editing tool that enables geneticists and medical researchers to edit parts of the genome by removing, adding, or altering sections of the DNA sequence. CRISPR-Cas9 system consists of two key molecules that introduce mutation into DNA. The two are an enzyme, which is the Cas9, and a piece of RNA called guide RNA. I'm going to draw some structures so you can further understand how the Cas9 system works. So right here, we have a DNA strand with a target sequence. The guide RNA consists of a small piece of RNA sequence about 20 bases long, located within a longer RNA scaffold. The scaffold part binds to the DNA, and the pre-designed sequence guides the enzyme to the right part of the genome. The Cas9 enzyme then binds to the guide RNA and acts as a pair of molecular scissors that can cut two strands of DNA at a specific location in the genome so that bits of DNA can then be added or removed. The guide RNA is designed to find and bind to a specific sequence in the DNA. It also has RNA bases that are complementary to those of the target DNA sequence in the genome. This means that theoretically, the guide RNA will only bind to the target sequence and no other regions of the genome. At this stage, the cell recognizes that the DNA is damaged and tries to repair it. Scientists can use the DNA repair machinery to introduce changes to one or more genes in the genome of the cell of interest. This system currently also stands out as the fastest, cheapest, and most reliable system for editing genes.